B cell maturation begins when you're an embryo, so fairly you're early on. The fetal liver and yolk sac and fetal bone marrow serve as locations for B cell maturation in the fetus. Then, after birth, the B cells kind of retreat back into the bone marrow, and that's where the primary location is for the maturation to occur. The graph or chart 11-1 in your book is a pretty good overview or representation of what can happen throughout the maturation process, throughout B cell developmental processes, where you even get your memory B cells and all. So that's just kind of a good quick overview of the whole process. Progenitor B cells are kind of interesting because these are kind of like they're not really pluripotent stem cells, but they are still in the embryonic phase. And they require interaction with bone marrow stromal cells, which is kind of like the yellow fatty looking material inside a bone marrow, inside a bone when you crack it open. So it requires that interaction. In other words, if we were to take these progenitor cells, the pro-B cells, and move them into tissue culture, we would not be able to get them to differentiate without the presence of bone stromal cells. There's some pretty interesting experiments done a number of years ago to prove that. Anyway, in humans they use in humans and in mice it's a tyrosine kinase, which helps to kind of trigger that. It's called CD45R in humans or B220 in mice. And it allows proliferation and differentiation of these pro-B cells into what are known as pre-B cells or precursor B cells. Now, what's happening inside these cells is they're going through some genetic rearrangements, and they're trying to produce viable receptors or antibodies. And if a B cell does not produce a viable antibody, then it will be killed and removed from the developmental process. How do I say this? If you have, let's say you need 4 legs to the table. You build one leg and you attach it to the table, and if it works, then you can proceed to the next leg. But in the case of the B cells, what they do is they'll build one leg and attach it to the table, and then they'll make 100 copies of that process, so that at the point where there's one leg on there, they've got 100 copies. That's one successful leg. And now each one of those copies will go through another set of differentiation or another set of genetic rearrangements, and they'll put a second leg onto the table. And then each one of those may have different legs in that second position, but they have the same leg in the first position. So you can kind of see that it's a slowly progressing thing, but they have checks and balances as they go through. Now, once the pro-B cell is produced and it's ready to start differentiating, it has a VLA4 protein on its surface that can attach to a VCAM1 on the bone marrow stromal cells. This kind of triggers production of c-KIT, which is another tyrosine kinase, on the B cell itself. Once both of these occur, then you start producing IL-7 receptors, that's interleukin-7 receptors on the surface of the B cell. And the connection also triggers the bone marrow stromal cell to produce interleukin-7, which is released. This causes the multiplication, the proliferation of more B cells at that point in time. So once you get to that point, then they'll start producing these pre-B cells, and then they'll start slowly developing and continue in their maturation process. See if I've talked about all of that. Yes, I've pretty much covered that. Next, we'll talk about immunoglobulin gene rearrangements and how they produce immature B cells.